Fun fact, every single cell in the body has the exact same DNA. It's super long and super complex. If that's the case, how does each cell in the body know how to be different? How does our heart cells know not to make protease, an enzyme found in the small intestine? How does our trachea know how to make cilia, and our skin know how to make hair follicles? All of this has to do with regulating transcription in each group of cells. Eukaryotes are much more complex than prokaryotes, as we know, so our methods are a little bit more complex than just operons. On the test, you will not be expected to memorize this, but just use them as examples of how our body regulates transcription. Most of the questions will give the information that you need and require you to apply your knowledge. There are 3 basic ways that eukaryotes can control how genes are expressed. 1. Eukaryotes can regulate how chromatin is coiled or uncoiled. 2. Eukaryotes can regulate when transcription starts. And 3. Eukaryotes have a way of regulating things after transcription as well, before translation begins or sometimes even after translation. Let's remember that prokaryotes do not have chromatin or tightly wound DNA. Chromatin is tightly wound DNA found specifically in eukaryotes. Chromatin can be further condensed into what's called heterochromatin. This is extremely tightly wound DNA, and therefore transcription factors cannot bind onto these areas. Euchromatin is not as tightly wound, and transcription factors can bind. Heterochromatin is DNA not expressed in these cells via transcription and translation. So, how do we regulate the chromatin structures? We can first modify histones. Histones are the proteins that actually keep chromatin wound up. If we add something called acetyl group to histones, called histone acetylation, it can loosen up the chromatin structure. If we take away acetyl groups, it can cause our chromatin to fold up tighter. We can also add or remove methyl groups. Methyls make the histones wind up tighter. Removing methyls via phosphorylation causes it to unwind. Methyl groups can actually be added directly to cytosine on the DNA as well. This does a similar thing that it does to histones, causing a reduction in transcription. Fun fact, histone modification can actually be passed down from one generation to the next. That means it's actually an example of epigenetics, since it shows heritability on things not direct with the nucleotide sequence. The next way eukaryotes can regulate transcription is literally with its initiation. Let's remember operons, structures in prokaryotes that can be regulated. Eukaryotes have similar structures, although we don't call them operons. Control elements are genes that serve as binding sites for transcription factors to start transcription. These control elements can have things bound to them to prevent transcription. RNA polymerase cannot bind without the transcription factors binding to the control elements. Control elements can either be proximal, which means close to the gene, or distal, which means far away from the gene it controls. We can call distal control elements enhancers as well. Genes can have multiple enhancers, but each enhancer is extremely specific to the gene it controls. Transcription factors can also help initiate transcription. Activators bind and enhance the rate of transcription. We also have repressors that can slow down the rate of transcription or stop it altogether, similar to what we talked about with operons. There are multiple combinations of activators, enhancers, and repressors that can control the rate of transcription in a eukaryotic cell. Lastly, we have ways after transcription that eukaryotes can regulate protein use. We've already talked about post-transcriptional modification and transcription. This only happens in eukaryotes in the nucleus. Post-transcriptional modification is altering the mRNA molecule to splice out introns and leave exons. Some introns are exons for other proteins, which means we can make multiple proteins from the same sequence of DNA. mRNA can actually be degraded before it reaches translation. Although eukaryotic mRNA lasts longer than prokaryotes since it has to leave from the nucleus, regulatory proteins can bind to the mRNA and cause it to degrade, blocking translation. One example of this is after egg fertilization in humans. Egg fertilization triggers initiation factors that stimulate transcription and translation, while simultaneously blocking other sperm from entering the egg, since we don't want multiple copies of different DNA in the cell. Proteins can also be processed to create different proteins. For example, after translation, proteins can get sugars or lipids added to them, like glycoproteins on our cell membrane. Proteins can also be degraded as needed, which is another example of protein synthesis control. Ubiquitination is adding a chemical, ubiquitin, to degrade proteins. Proteasomes are complexes that degrade proteins as needed as well. Finally, let's look at how non-coding RNAs can control gene expression. Only about 1.5% of our DNA holds the code for proteins. Wow. If that's the case, what's the rest of the DNA for? This non-coding DNA codes for non-coding RNAs, or abbreviated ncRNAs. All of these RNAs regulate mRNA creation and translation, as well as chromatin configuration. We call it non-coding since it does not directly code for proteins. MicroRNAs, or miRNAs, are single-stranded RNA molecules that can bind to mRNA. They signal for the degradation of mRNA to stop protein synthesis. RNA interference caused by small interfering RNAs, or siRNAs, also block gene expression. siRNAs can also affect heterochromatin and can block parts of the chromosome from being transcribed. There's a little cute type of RNA called piRNAs, or piRNAs, that can induce heterochromatin structures, meaning it winds the chromatin up tighter. Please do not stress about the different types of RNAs, as I've never seen a question about these. But I thought I should talk about them since they are DNA sequences that don't actually code for proteins. They code for RNAs that regulate our mRNA. That's probably the biggest takeaway about this.